Good morning. Morning, everyone. Hi, good morning, everyone. With the dangerous task of interrupting breakfast, it's really a great pleasure to welcome everyone here today. My name's David Anderson. I'm the Dean of the Faculty of Medicine at Dalhousie University, and it's a pleasure to have you joining us here at our Breakthrough Breakfast to celebrate research excellence. In a few moments, we're going to hear from members of our immunology, inflammation, infection, and vaccinology research team, better known as I3V. Before we begin, I would like to recognize that Pier 21 is located in Mi'kma'ki, the ancestral and unceded territory of the Mi'kmaq. We are all Cree people. We also recognize the histories, contributions, and legacies of African Nova Scotian people who have been in these lands for over 400 years. I'd also like to acknowledge the significance of Ramadan and extend our warmest wishes to those celebrating. With this in mind, we have arranged for takeaway boxes for those participating who wish to bring some pastries home to enjoy after sundown. So again, it's great to be able to get together in person today for relaunching our Breakthrough Breakfast. These breakfasts are a way to let our generous community supporters know about about the fascinating work that's happening amongst research at Dalhousie. Work that's changing lives and that wouldn't be possible without donors and partners. I would like to acknowledge our sponsors for today's exciting event. Thank you to our gold sponsor, Shanax, and to our bronze sponsors, Sobeys National Pharmacy Group and Innovative Medicines Canada. A bit of housekeeping. I'd like to remind you that back behind us in the pavilion, you'll see a table with the latest copies of Remedy magazine. It's filled with stories and highlights of research that's happening here at Dalhousie and demonstrates the impact of philanthropy supporting our innovative work. Please take a copy home with you today. As Dean, I'm inspired by the passion and dedication of our faculty members, researchers, and students. They push boundaries of scientific inquiry every day. Their innovation produces new treatments, new approaches, and results in better healthcare for all of us. Health research is a team sport, and we gratefully acknowledge the support of collaborators at Nova Scotia Health, the IWK Health Centre, the provincial government, industry, and the people of our region, all of whom play important roles in Dalhousie's health research success. Today's group of researchers focus on areas that are critically important in today's society. Again, focused on infection, immunology, inflammation, and vaccinology, this team is uncovering ways to help our immune systems fight everything from coronaviruses and the flu to cancer and lupus. The discoveries our Dalhousie researchers have made have changed the way we treat, prevent, and manage scores of illnesses. To move a discovery from the laboratory to the front line of healthcare, the clinic, and to your family doctor requires biomanufacturing facilities and expertise the Atlantic region currently doesn't have. Our team has been working very hard with stakeholders across the medical research and healthcare continuum to change this with a vision to establish a new medical research facility that will, amongst other things, help bring homegrown discoveries to life. Stay tuned for exciting news about this project in the months ahead. The impact we're having here in Nova Scotia will change lives, and by investing in medical research, we're building healthier communities, not just for us, for generations to come. Our donors have an incredibly important role to play in our success, and we thank you. So thanks again for coming this morning. I hope you enjoy your breakfast and the presentations. And now it's my great pleasure to welcome Dr. Kim Brooks to the stage. Kim is our President and Vice Chancellor at Dalhousie. Thank you all very much.
All right, good morning, everyone. You all look awesome. Let me say that I know at these kinds of events you want to listen carefully to the researchers who say all the important things, but we welcome the clanking of dishes. We want you to eat and be fed and sustained. And so eat, eat, eat. Don't be reluctant to make those noises. Reach across, grab a pastry. As David mentioned, I'm Kim Brooks. I'm delighted to welcome you to the return of the Breakthrough Breakfast. I'm a lot shorter, it turns out, than David. So I'm wrestling a bit with the microphone, but I think I've got it. Today we're celebrating innovation and discovery in medical research at Dalhousie's Faculty of Medicine. I want to thank everyone for getting up early and for joining us to dive into what is going to be a fascinating conversation about some of the research that's going on. I want to thank David in thanking Shannix, our gold sponsor, and Lawtons and Innovative Medicines Canada, our bronze sponsors. That support, as you all know, is critical to helping us host these breakfasts where we are able to bring people together so you can hear about some of the great research that we're doing. Today, our three exceptional researchers will give us a snapshot of their work in immunology, inflammation, infection, and vaccinology, which, as David mentioned, we call the I3V group. Their dedication, passion, and relentless pursuit of excellence is inspiring. Before we hear from them, I want to echo David's excitement about collaborating across the region to invest in a biomanufacturing facility. It's an investment that will fuel leading research and ensure that our best discoveries can be shepherded through the development process to ultimately improve health outcomes. The new facility will not only fill a critical gap in the research pipeline in Atlantic Canada, it will add much needed capacity to Canada's entire life sciences and biomanufacturing sector. We learned during the pandemic that not having enough manufacturing capacity for new drugs and vaccines is a critical weakness. Our new facility also underscores Dalhousie's commitment to translating the medical research done in our labs into real, tangible healthcare solutions. Please sit back, eat your breakfast, and enjoy listening to these three IV3 researchers about their research. If you have questions while you're listening, you might jot them down. We'll have a question and answer session afterwards. We're going to start. I'm sorry you didn't get any breakfast. By hearing from Dr. Lisa Barrett, someone you will recognize well if you spent any time in Nova Scotia over the last few years as she rolled out Nova Scotia's rapid COVID testing program. Dr. Barrett is a clinical investigator, assistant professor in the Division of Infectious Diseases, and principal investigator of senescence, aging, infection, and immunity, SAIL, laboratory. She studies immune responses in chronic and persistent viral infections, and she is well known to this crowd. I invite her to the stage.
Well, I haven't felt that much pressure to eat since I've been in my grandmother's kitchen in Newfoundland, but please do eat and eat well. Can you folks turn the mic down a little bit for me in the back? As I said, I'm pretty loud. Can folks hear me okay when it's down a little bit? Great. Okay. So I've given myself 8.5 minutes to tell you about aging and immunity and why it is so important for everyone in this room. And President Brooks is going to be giving me the countdown because this is not something that you should think of in terms of contributions, in terms of research that should happen later. It's now. So she's going to create that imperative for us. Goals for aging. Someone shout one out. Anyone have a goal for aging? Independence. Mobility. It's a good one. Oh, social life. Oh, okay, I can't quite hear everyone, but I hear a lot of things that have to do with A, being alive and on board. That's important. You got to be around to do any of those things. And number two, being able to do that well and having enough health to be able to do that. Did you know that even today, with all of our amazing advancements in vaccine research, that we still have infections in the top 10, in fact, in many countries, the top 5 reasons that people over the age of 70 die. So immunity clearly in terms of infection hasn't been sorted out in older people. And in fact, it hasn't really been a focus of research in many domains over time. Why? How do you consent people? They may have dementia. Do they want to be bothered with research? Do older people want to be included? And so there have been many barriers over time that mean we don't fully understand what it means to have immunity to different infections or to prevent them as you get older. And that gap, as you might imagine, was a very, very big problem when you a novel pathogen such as SARS-CoV-2 or COVID-19. I think at some point we're going to drop the 19. So with COVID, that was going to be a challenge. And we noted, especially in Nova Scotia, of course, our wave one. Remember back to April of 2020? I don't want to, and I'm sure you don't either. But in April of 2020, most of our wave one deaths in Nova Scotia happened in long-term care. The vast majority in older people. Was that atypical or different compared to other provinces? Not entirely, but neither was it quite as regulated or as distributed as it was in other places. And we know that Nova Scotia has a population that's aged, and there are other medical conditions along with it. But we don't really understand a lot about the nuances the nuances of what makes us have immunity or not, do well or not. It's easy to say we have these broad sweeping strokes that we apply to older folks, right? Oh, they have lots of other illness, this big swath, lots of other illness. Well, frankly, lots of people with lots of illness do quite well and don't get infected every day. So clearly, there is what we would call in the world of research a difference in the phenotype between individuals. So, a couple of gaps. One, we weren't quite sure with COVID coming how people who were older were going to respond, how they were going to respond to vaccines. And for those in the audience, and many titans of immunology in this audience, for whom I have a great deal of respect, we didn't often have an opportunity at a broad scale to look at the response in older people to a novel pathogen. So we're always looking at how people respond, how their immune systems respond, T cells and B cells, we're going to talk about those in just a moment, how they respond to things they've seen before as younger people, influenza, common cold, other viruses, other infections. We almost never have the opportunity at a broad scale, thank goodness, and not looking for another one. Please hold back the measles for at least a few more years. We had an opportunity with COVID to say, how will people respond to a completely novel pathogen, a virus, in older folks in long-term care? And hence LifeCo. So in term care in Nova Scotia, in the first wave at Northwood primarily, and some of its affiliated facilities, we enrolled 108 people and followed them at 30 days, 4 minutes. And you feel that? You feel that impetus? That's what we felt the entire way through that whole wave. Because we needed to get to people. We wanted to be able to see folks before they got COVID, during COVID, after COVID, and see what their immune response did. And when I say immune response, I'm going to talk specifically about T cells, and we're going to speed up now because things speed up, time speeds up. We don't have time sit around and not know this information anymore. So we were looking at B cells and T cells. They're the parts of your immune system. They float around, among other places, in the blood. We went to the long-term care facilities. We got consent from people to be involved in research. 0 human beings said no to being involved in this research. We went back to our lab, and then we assessed the T cells and the B cells for their response to this novel pathogen immediately and in the memory phase after 30 days and 8 months later. And you know what we found? A couple of things that were really interesting, and one of which is very interesting. One, people who are older can mount pretty good responses, and we won't talk about the vaccine part of that study because we'll leave the vaccine work to Dr. Halpern for later. But one, people can respond, but not everyone does. And in fact, up to 40% of people didn't have great T cell responses, and a good chunk of those folks didn't have responses at 8 months later that were even detectable. Does that mean that they were susceptible to infection again or that they were sick every day? No, but something more subtle. How might they respond to a vaccine? And were they going to be protected as well as someone younger or who had different T cells later on? We don't quite know the answer to that yet, but we should, because if we know the answer to questions like that, we can build better vaccines, not just for populations, but for the people who were the non responders. And so, with the piece of information that we found was that people who had an infection with something we call CMV, cytomegalovirus, about 50% of the people here have it. It sits in the background of your immune system, but it's a space occupying lesion. Do you know what? Those people had very exhausted T cells and were far more likely to not have durable memory responses. Is that something we can change? Maybe not, but maybe we could start to develop better vaccines, better preventative and better treatment options that'll integrate that information. And we didn't know that before. Thank you to all the Nova Scotians who actually contributed to that. So, what do I want you to take away from this? One, there is a huge knowledge gap in older person immunity. And with those folks being the people who die the most of infections in our populations, we need to reprioritize them both with funds and with education to enable that research to happen quickly. And number two, keep going because we can learn something, and people who are older want to be involved in research every single day. Don't leave them out. And so, for mobility, for a social life, to be present, to enjoy friends and family, we can do better. And we're going to do it better. And at Dalhousie University, in the immunity department and in I3V, we have the ability and capability to keep doing that every single day as long as we keep prioritizing older people. Thank you for your attention. Thank you for coming this morning. And remember that those people who were in that Wave 1 research of LifeCo that you can see here, we're in the middle of a pandemic. They've agreed to show these pictures. Every single one of these humans have contributed to some of the first novel pathogen, oldest person, greater than 100-year-old people on the planet, consolidated research that's being put together with a Canadian database in the world. So Dalhousie is leading here. Nova Scotians are leading, and their kindness I will never forget. Thank you very much for your attention.
Thank you, Dr. Barrett. You gave me a sense of urgency anyway, and now I feel like the rest of my day is going to be supercharged. I'm going to invite to the stage Dr. Scott Halpern. Dr. Halpern is a professor in pediatrics and microbiology and immunology and the director of the Canadian Centre for Vaccinology. Dr. Halpern's area of expertise is whooping cough, and his research focuses on the diagnosis, treatment, and prevention of vaccine preventable diseases. Please welcome to the stage.
So it's a bit intimidating always to follow Lisa Barrett as speaking. In fact, I had encouraged them to put me first because there's an old adage that says that people are happiest and you're perceived as being a much better speaker if people are eating at the time, and even better if they're drinking, but a little early in the morning for that. So instead, they decided they'd sandwich me between the two good speakers, and hopefully I'll settle things down a little bit, bore you a little bit, and then Craig can initiate things again. So I'm going to talk about vaccines, and that's sometimes a bit controversial. There may be some people in the room who don't believe in vaccines, and unfortunately, I'm going to then offend you, and I'm not going to apologize about that. Vaccines have been and were identified by the U.S. Centers for Disease Control as the #1 public health achievement in the 20th century. And in that citation at the end of the 20th century, they cited things such as the complete eradication of smallpox, the near elimination of polio, control of multiple other vaccine-preventable diseases, and the prospect of control of several other diseases that were new vaccines were just coming to the fore. And in the past 20 years of the 21st century, we have controlled those diseases as well. In fact, if you look at the numbers, you can see that from, in some cases, hundreds of thousands of cases, we've eliminated down to a handful of cases. Now, that's not the case in some diseases like pertussis, which is also called whooping cough, and I'll get into that a little bit more. You can see a remarkable decrease from 150,000 cases at the beginning of the 20th century to only 3,000 cases. These are in Canada at the beginning of the 21st century, but it's not as good as some of the others. Polio is a remarkable achievement. This is a picture of iron lungs, of wards of iron lungs in the 1950s, where people who were paralyzed from polio were in iron lungs to help them breathe. And it's nice that we're not seeing that anymore. In fact, there are 3 different polio viruses, and we have been able to eradicate 2 of them. So the first one, which is poliovirus 2, was eradicated in 1990, and 30 years later, in 2020, poliovirus 3 was eradicated. And all that's left now is poliovirus 1, and there are only 2 countries left, Afghanistan and Pakistan, where there's actual endemic spread. Now, there are cases of polio elsewhere. People who are traveling from Afghanistan and Pakistan to their home countries where they manifested. There's no transmission of polio anywhere else except those 2 countries. And hopefully, we'll be able to eradicate the 3rd and final polio strain, and polio will go the way of smallpox to never be seen again. Now, whooping cough is a disease that we haven't been able to get rid of. And if somebody can play the video. This is a child with whooping cough. And as you can see, it's not a pleasant thing for this child. You can hear the whoop at the end of a bunch of coughs. Now, whooping cough, well, that sounds miserable, and it can go on. In Chinese, it's called the 100-day cough. It can go on for months. In younger infants, it can actually be fatal. And in fact, in Canada, we still have a half a dozen deaths from a whooping cough every year. And it's in those very young infants who are too young to have started their immunization series. We want to have a better whooping cough vaccine. There's some vaccines like measles, which are 95% effective. And because measles is the most infectious virus we know, if you walk into a room and you have measles and there are 3 people who weren't immunized, those 3 people are going to get measles. It's that infectious. The measles vaccine, if you get it and you get your 2 doses, it's lifelong immunity, just like the virus itself. So why are we having outbreaks of measles that you've read about over this past several weeks? Quebec, there have been actually transmission of measles here in Canada, which we hadn't had in over 30 years because we had eliminated transmission of measles in Canada, and it was just imported cases. But now we're having some transmission. It's not a failure of the vaccine, it's failure to vaccinate. And that's one of the effects of the COVID pandemic is people think we're tired of vaccines. Well, that's great, but the infectious diseases are not tired. They're still there. And now we're seeing measles back. And that's what Lisa was mentioning. We don't really want to see a pandemic of measles again. That's something we don't want to see. That's a deadly virus. Pertussis, the vaccine isn't as good as the measles vaccine. So even people who are immunized, the immunity doesn't last forever. In fact, you can see we get doses of the pertussis vaccine at 2, 4, 6, 18 months, preschool, and again in adolescence. And after you've had the vaccine, within several years, that protection wears off. and we give a booster in adolescents, and within 2 years, people are susceptible again. You can see the number of cases going back up. So we need a better vaccine. So what I'm going to talk about is, I named this the alphabet soup. The first letters are CCFV, or the Canadian Center for Vaccinology, and that's part of the I3V group. And we have over 30 or so investigators at Dalhousie as part of the group. It's a collaboration between Dalhousie, IWK, and NS Health, and over 100 staff and scientists who are working with us. So the group is about 150 people strong. It's 3 groups. There's a discovery group, which is basic scientists looking at how vaccines, developing new vaccines and figuring out how they work. The evaluation group that does clinical trials. We're very active in doing clinical trials for the new COVID vaccines during the pandemic. And then the policy program and implementation group, which is a diverse group of scientists, social scientists, that look at how vaccines are accepted in the population, vaccine confidence, vaccine resistance and hesitancy, and also how programs work and the effectiveness of vaccine programs. CCFB is one of the areas we're working on are new pertussis vaccines. And this is a challenge unit. So what is a challenge unit? So this is one of only 2 units in Canada and only a handful worldwide. And this is a unit at the IWK of 10 beds with full isolation capabilities. And in this facility, we can do something called a CHIM, more initials. A CHIM stands for a Controlled Human Infection Model. We're recruiting here if anybody wants to do it. So let me tell you about CHIMs. So CHIMs are where we take an infectious disease that we're wanting to study, and we give it under very carefully controlled circumstances, give that infection, whether a bacteria or a virus. In this case, we're talking about the pertussis bacteria. We give it to volunteers, healthy volunteers, who are then admitted to this unit and monitored closely for up to 3.5 weeks. And what we're doing is looking at the development of that disease, not the full pertussis that you saw in that case, but the early symptoms. And once they have the early symptoms, we treat them with an antibiotic, cure the disease, and because if you treat it early, it doesn't progress on to what you saw in that young infant. Why are we doing that? Well, one of the reasons is that under those carefully controlled conditions, we can actually determine the immune response to the bacteria. And by understanding better the immune response, we'll be able to develop better vaccines. Another thing we can do in this unit, and that's something that we're going to be doing, beginning a study this summer, is to test a new vaccine against whooping cough in individuals. So we can give a vaccine, a new vaccine to some people, and randomly give a placebo to others, and then challenge them with the pertussis bacteria, and then monitor them closely and see who develops those early signs of pertussis and who doesn't. So does the vaccine work? And that can short circuit something, because in other circumstances without a challenge, one would have to go out into the community, give a vaccine, give a placebo, all the things that you heard about at the early part of the pandemic with the COVID vaccine, and immunize over 40,000 people, as we did for COVID, and then monitor them over time to see if the vaccine works. Now that worked with COVID because COVID was everywhere. Whooping cough occurs in cycles, and you can't predict when the outbreaks are going to be. So you don't know who to immunize, because you can immunize 40,000 people here, the outbreak can be somewhere else. So the CHIM study will allow us to test new vaccines against pertussis and hopefully then bring these to people. So that gets to the next couple of letters of GMP. And what is GMP? GMP is good manufacturing process. And this is a mechanism, a method of manufacturing, whether a drug or bacteria or a virus, under very strict conditions and controlled conditions, so that you know exactly what you have and it's reproducible. And that is required to get it approved by the regulatory authorities, because safety is the most important thing. For chimp studies, we need to give people that organism. So how do we do that? Well, back when I was first uh, doing research, we were looking at rhinovirus, and the way that was done was you got somebody with a rhinovirus cold, took some of their snot, sorry about the breakfast, and you took it, you aliquoted it out into small amounts, and then you gave it to other people. Okay? Very simple, and those people then developed a cold, and you studied those people in a challenge unit. Problem is that between that time and now, things such as HIV and hepatitis C were understood, and you can't do that anymore because that wouldn't be safe. So now you need to manufacture that organism under GMP conditions. Now for pertussis, because we can grow pertussis in completely defined media, so just chemicals, no blood products, no human products, no cells. For pertussis, we are able to do that in a GMP-like facility where everything is done the same way as GMP, but it's just not a facility that was built specifically for it. This is a laboratory at the IWK, which we modified to be GMP-like, and we were able to manufacture the pertussis bacteria in order to use in the chimp. You can't do that for viruses. For viruses, and that has to be in a GMP facility, which is why we are going to be building a GMP facility. And this is some pictures of a laboratory here in Nova Scotia that we're planning to modify into a GMP facility. And hopefully within a year or two, might be a little optimistic in terms of the timeline, we'll have a GMP facility that where we can do GMP manufacturing of virus and bacteria. That's not alone the only thing that we can do with it, and that's why it's got the capability of doing far more. I'm now going to venture way out of my comfort zone. One of the other things that it can do is do a therapy called CAR T therapy, and Lisa will take any questions about this, but CAR T is called chimeric antigen receptor T cell therapy. And what that is that one can take a patient's own T cells, modify them in the laboratory by putting another receptor into them, which then allows those T cells, a person's own T cells, to target a specific cancer cell. So it's a personalized medicine method of treating cancer therapeutic. It's been highly effective. We don't have that capability of manufacturing those here in Eastern Canada. So this type of facility will also allow us to do that type of research and therapeutic. I mean, that's bedside treatment. And it will allow us to do other things as well. It'll allow us to also to make proteins for potential new vaccines, antibodies that can be used to treat infections. So a GMP facility has a wide variety of potential, and it will be a real put Nova Scotia on the map in terms of being able to do the type of research that we need to do to have innovative therapies, innovative treatments, and innovative preventions. Thank you.
All right, thank you, Dr. Halpern. Our final presenter today is Dr. Craig McCormick. He's a professor in the Department of Microbiology and Immunology, and he co-founded the Canadian Society for Virology, CSV, and co-founded and served as director for the Infection, Immunity, Inflammation, and Vaccinology team at Dalhousie. Howdy. Dr. McCormick is an expert on host antiviral responses and viral evasion mechanisms, focusing on emerging and reemerging viruses with pandemic potential like influenza A viruses and coronaviruses. Welcome.
Very good. I'm really pleased to have the opportunity to be here with you today and tell you a little bit about what we do and our approach to research. As I'm a molecular virologist, so What we do is we study viruses at the molecular level, think about genes, proteins, viral genomes, and we try to understand them better in order to develop new vaccines, use this information to develop antivirals, interventions, and hopefully understand where viruses come from, and if we can stop their spread and prevent future pandemics. I'm looking out here, and I just wanted to mention my grandmother was a war bride who came through Pier 21 80 years ago, and I think that she would be very pleased that I was up here speaking today. So. So she basically landed here, hopped on a train up to Moncton to live with her mother-in-law in New Brunswick for a couple of years and wait for her husband to come home. So it was a time, it was kind of a dry province at the time anyway. Uh, it was a hard time for her. Um, so yes, molecular virology. So my laboratory provides a training environment where young people get an opportunity to learn about how to study viruses safely and learn about their properties. Um, to do so, so basically what they do in layman's terms is they take viruses apart and figure out how they work. Um, and they use a tactic that's a common tactic in science education called deconstruction. So imagine a broken clock. So I come from the kind of family where if the clock was broken, we would change the batteries and then we were out of ideas. Except for my uncle Albert, who was an engineer, and he would love the challenge of taking apart a clock and putting it back together. And so much so that he actually had a side business in repairing score clocks for local rinks in Ontario. So that's the principle of deconstruction, uh, taking something apart to learn about it. So I learned about this tactic of deconstruction to study viruses early in my training. So this was as an undergraduate 30 years ago at the University of New Brunswick. I studied a very simple virus called a bacteriophage that infects and kills bacteria. And the simplicity of this infection model really made it a darling of molecular biologists sort of in the 1960s and 1970s. What they would do uh, is they would basically take these viruses, interrupt genes one by one, create collections of mutant viruses that had gene deletions to figure out, and then basically test them, challenge them, and see what their properties were like. Actually brought it wrong. So whenever I look at a glass test tube, it reminds me of the 1970s era yellow GE refrigerator in our laboratory that was full of racks and racks of these, and they appeared to be filled with skim milk. But it was a situation of don't drink the forbidden skim milk because it was about 1 trillion bacteriophage. There would be normal bacteriophage, and there would be the ones that were broken in key ways that allowed you to study them. So this is this idea of deconstruction. So today, so my laboratory has postdoctoral fellows, graduate students, and then younger trainees who sort of learn from the postdocs and graduate students. And there's a hierarchy and mentorship that goes on in order to basically train people to work with viruses once again safely, but also hone their critical thinking skills and communication skills. Um, so just one example here. So Alexa Wilson is one of these sort of like peer mentors for junior trainees in the lab and kind of a role model. So Alexa's here with us today. And so what she does is she studies a virus that causes cancer. Um, and so the challenge for her is to figure out, okay, how does this virus evade the immune system, evade our sort of surveillance system, and how does it set things in motion to initiate cancer? Um, she uses the principles of deconstruction. So what she'll do things like investigating the function of a single viral protein. So let's break it down into component parts. How does this individual viral protein work? Does it have immune evasive properties? Does it have cancer initiating properties? Then what she can do is use deconstruction in a different way. Once again, sort of like I did 30 years before, taking viruses that are lacking her gene of interest and comparing them to normal viruses. And by that, that compare and contrast allows us to learn about their properties. But yeah, how does the broken virus fail, basically, is the question. Okay. Because this is 2024, this could be a picture of Alexa at the microscope. She's been doing that a lot lately. Because it's 2024, what she does is she learns something, and instead of keeping it to herself for a while, we of course have a lab messaging system where everybody shares data as sort of with the group live. And so my phone pings and I get pictures of what she's seeing on the microscope. And this kind of expresses the enthusiasm you have for that moment where you make a discovery. So, so in the spring of 2020, like virologists all over the world, we learned to work with coronaviruses and to participate in the pandemic response. Um, this work, the early part of this work was really led by two very talented postdoctoral fellows, Eric Pringle and Brett Duguay, and these are other members of the team. And the goals were basically to learn to work with the virus, and sort of by being first in the region to do so, it put us in a great position to be able to collaborate with others to test antivirals, vaccines, personal protective equipment, PPE. Um, so our goals were basically to establish research capacity, um, to investigate, see what we could learn about the fundamentals of how coronaviruses work, using those principles of deconstruction. And then, like I say, collaborate with others on vaccines, antivirals, and PPE. So what did we learn along the way? Um, Okay, so I've got a lot of enthusiasm for this particular project. We did learn one thing sort of through deconstruction. We learned that these coronavirus M proteins. So I've got a cartoon of coronavirus here up at the top, and you can see, you've probably all seen this cartoon actually. Um, so you've got the spiky projections in blue, and then there's this protein in red that's around the periphery. And this protein is very important for bringing the component parts of the virus together and assembling into an infectious virion. Basically, it makes contact with the genome in the inside and spike proteins on the outside. Okay, so it's a structure, it has a structural role. That's how the world understands this protein. But this red protein, so Dr. Eric Pringle and PhD candidate Taylor Cadell were working on this particular protein and found that it had basically the ability to inhibit host responses to infection and to stress. And so they're trying to understand exactly how this works. They're investigating molecular mechanisms. What's interesting about this new property of like formerly just something that was known as a structural protein is that it's broadly shared amongst all the different human coronaviruses, including the ones that circulate among us and just cause mild disease. So we're trying to understand this broadly conserved ability of this protein to inhibit host responses to infection. Okay. A second project I just wanted to share quickly with you was around antivirals. We had, at the time, just at the start of the pandemic, we had been working with this antiviral drug candidate that we knew could inhibit influenza virus infection. And so we naturally wanted to test it against coronaviruses. What we found is it was quite effective, and it had a very interesting property. So I've got the cartoon of coronavirus here again, and you can see the blue spiky projections. And so what we learned was that this particular antiviral drug candidate seemed to interfere with spike specifically. So much so that you ended up having basically, so here are normal coronavirus virions with spiky projections by electron microscopy, and see that the drug-treated ones sort of bald and spikeless, and therefore non-infectious. So we thought that this was a pretty cool phenotype, and we're following up on exactly how this works, and in hopes of making better drug candidates that could have similar properties. So we recognize. So we work with viruses, we're microbiologists and immunologists. We don't have exactly the right expertise to do antiviral drug discovery. We need to tap into different expertise, chemistry specifically. And so we're very lucky at Dalhousie to have great colleagues who work in this area. It's Dr. Sultan Darvish and Dr. Ian Potty, and we've been collaborating basically to use modern principles of how people do drug discovery with regard to computer modeling. So what you can do is you can take uh, a target protein that's being bound by a drug. The stick model in the middle is your drug. Details don't matter. But basically, what we can learn is precisely how a drug fits into a pocket. And then we can use that structural information to make predictions about how to make a better drug that fits into the pocket better. Imagine it having greater selectivity, better efficacy, probably better safety, the more targeted you could make it. And so we're very excited to collaborate with chemists. We speak very different languages, so this is virology and chemistry that we teach each other a lot in meetings. Really looking forward to where this work goes. Okay, I think I'm going to get the 8.5 minutes. So I just wanted to close on two thoughts. One was obviously, this presentation is very centered sort of on the trainees who do the work. And I just wanted to, this opportunity to speak with you today gave me a chance to kind of reflect back on the people who have trained in my lab. And so I created a word cloud, which is very out of fashion, but I created a word cloud. And so what it made me think about was the people that have trained in this research training environment. Some of them have gone on to be professors with laboratories of their own, doing their own kinds of research. But many others have become physicians. Working in public policy, lawyers working with intellectual property, and I'm very proud of them. And they've also kind of in many cases have used their strong foundation in research to sort of continue on through their careers. So I just wanted to put this up here to sort of highlight that we're not just training people to work only in this sector, but there are broad societal benefits to the training that we provide. And then lastly, I just wanted to talk about, so the students I'm training right now, this is an incredibly exciting time for us because there are significant investments happening in pandemic preparedness and in the biomanufacturing sector. And this means more jobs for our young people. And I know that they're excited to be able to contribute to the economy in this way. And thank you very much for your time.
Thank you. Okay, I'm in the awkward position of trying to bridge our commitment to letting people leave at 9 with our commitment to letting people get a chance to ask a couple of questions to our great researchers. And I think how I'm going to do that is to suggest that the researchers who are sitting near the front of the room, if you don't mind staying till just shortly after 9, will give people a chance to wander up and ask their questions to you before they leave. And I will draw this morning, I think, to a close. Let me say a couple things as I do that. One is thank you so much for coming. The second is my grandma also came through Pier 21. At least that's what she told me. And when I got here, I went down to the research center, which is spectacular, to get the ship manifest. And it turned out she went through New Brunswick. And I said to her, Grandma, you went through New Brunswick. And she said, No, I didn't. I went through Pier 21. And I said, Yes, that's right. Please take home your breakthrough breakfast mugs. There are flowers on each of the table. If you'd like to take them, carry them with the beaker into the atrium area and we will wrap them for you. Thank you so much for coming. To the researchers who were able to come and talk to us a bit about what you're doing, it is so inspiring and amazing, the work that you're doing. It makes me so proud to be a Nova Scotian and here at Dalhousie. And so you have our collective thanks for sharing your wisdom with us this morning. To everyone else who came and spent a morning with us, please do it again. The next one is June 25th. We look forward to seeing you there. And if you have a burning question or two, please use this couple. I think we've got 7 minutes left before the 9 o'clock close. Please use that time to grab someone who is offering us some of their wisdom this morning and ask your question. Thank you very much for coming and have a great afternoon, morning, afternoon, and evening.